Hello, everybody. I'm very glad to participate in this Simon Searchlight Family and Research virtual conference. My name is Reza Asadullahi. I am a geneticist working in the Institute of Medical Genetics, University of Zurich, and I'm going to present an overview of the MET13L variants and clinical features which have been reported in the scientific literature so far. You may know that MET13L syndrome was first recognized in these two lovely girls and published in 2013. And during the last 8 years, there have been 110 cases reported in the literature with disease-causing variants affecting MET13L. We know that MET13L is now among the top diagnosis in patients with intellectual disability. In this graph, you can see MET13L here. It is among really the top genes which is detected to be mutated in the diagnostic labs. Before I go through the variants and clinical features of MET13L syndrome, I want to remind you of a central dogma in biology, and that is the DNA, or our genetic material, is first transcribed to RNA in the cells, and then RNA is translated into protein, and proteins are basically the complex molecules which are crucial for development and functions of cells and organs. So they are the executive molecules, in fact. In normal situation, this process happens a lot in our cells. However, if variants affect our genetic material or DNA, they can have consequences on the protein. Based on the consequences of the variants on proteins, we could very roughly categorize the variants into 3 different groups. The first group of the variants are called truncating variants, and these are the variants when occur in the DNA, and they are also going to the RNA, and they lead to abnormal protein, a protein which is, for example, smaller than the normal protein, or no protein is produced at all. These are called truncating variants. Another class of variants are called missense variants, and these are those that the variant at the DNA level leads to only one amino acid change in the protein. If this amino acid is playing a very crucial role, then this can lead to a disease. And the third group are called gains, and these are those variants that lead to the overexpression of the protein. Basically, the protein is normal in the structure, but is produced more than normal. If we consider these 3 groups of variants and want to see what is in the literature based on the percentage of the variants, we can observe that the majority of reported variants are from the truncating group. 73% of the MET13 variant are truncating variants, which lead to either abnormal protein or no protein at all. Secondly comes the missense variants. 25% of the patients basically have these type of variants, which affects one amino acid. And third are the gains, which leads to overexpression, and these are about 2% of the reported cases. What I want to emphasize here, however, is that these variants are affecting only one of the 2 copies of MET13L gene, because this is a dominant disorder and only one copy is affected. The other copy is normal and produces normal protein. Here in this slide, I would like to show you a very simplified and schematic representation of MET13L gene, which is here on the top, containing 31 exons, or the units of the gene, which are vertical lines here, and then the structure of the protein, which contains 2,210 amino acids, which are also the units of the protein. And here you can see these arrows, which are indicating the location of missense variants affecting MED13L in the literature reported. You can appreciate that most of the missense variants are clustered in two specific regions, here and here. So one region is between the two so-called LXXLL motifs, and the other hotspot is the C-terminal domain of the protein. This presentation basically shows that these parts of the protein are quite important, that even change of one amino acid can lead to the disease. However, the lower percentage of reported missense, which is 25 versus 73%, which was for the truncating, is in part due to the fact that we still don't know the structure of the MED13L protein very well, and therefore sometimes we cannot classify missense variants which are detected in the diagnostic labs. And these missense variants are usually called variants of uncertain significance, and we basically don't know if they are benign or pathogenic. And that's why if you would have better tools in the future, probably the number of missense variants will also rise. The other fact that I want to mention is the pattern of inheritance for the reported variants in the literature. There are also 3 patterns of inheritance observed. The first of them is basically called heterozygous de novo variants occurring in the patient only. In this case, the father and mother are completely healthy. They don't carry any mutation, and the mutation occurs in the child newly, and these are called de novo variants. 92% of variants are in this category. However, there are few cases reported in the literature that are following the phenomena so-called mosaicism. And mosaicism in this case means that the parents are completely healthy. However, some of their germ cells, sperms in the father or eggs in the mother, are carrying the mutations or variants. And these variants are in a mosaic state in the parents. They create no problem for the parents, but in the germ lines, they can be transmitted to the offsprings, and then even more than one child can be affected. The third pattern of inheritance that has been reported is that the variant affecting MED13L is inherited from a parent, and this is the case that the variant causes very mild problem, and this is only reported for the gains of the MED13L or overexpression of the protein, because this is a very mild situation, and then the mother is mildly affected and have the variant, and this variant is then transmitted to the child. Among the 110 variants in the literature, not all of them are together with detailed clinical descriptions. Only 91 of these 110 have been with clinical descriptions, which I'm going to also show you briefly in the next slides. Basically, from the gender point of view, there are almost 50-50 male versus female. And in this graph, you can see the age distribution of the patients. The majority of patients are in between 16 months of age to 15 years. And we are basically missing information afterwards. There are a few cases in later ages. However, the data in this age range is completely lacking. And this is also one of the things that we need to know more about in the future. The main features from the reported cases are summarized in this graph. As expected, the majority of patients have developmental delay, speech delay, and motor delay, which you can see 99 and 98%. They're blue here. The blue means the feature is present. The green means is absent, and the gray or NA is not available. So the clinical data was not available. And you can see after the speech and motor delay, hypotonia or low muscle tone is the third. 60% of the patient present with that. 37% have abnormal brain MRI, but the abnormalities are usually minor. 25 or 24% of the patients have autism or autistic-like features. In 20%, there have been seizures reported, and 18% of the patients have had ataxia or coordination problems. When we look more into the details of the speech delay, it is evident that the majority of patients have moderate to severe speech delay. However, and there are some patients with mild, very few, and also cases with normal speech. And I will explain you what's the reason for that in the next slide. For the motor delay, and the majority of the patients have mild to moderate motor delay, and the most available data in this regard is about the age of walking, which is the mean age is 27 months. And this also clearly shows that the motor delay is less severely affected than speech delay in MED13L patients. I mentioned in the previous slide, and also when I was talking about the pattern of inheritance, about the gains. And if you remember, the gains are those variants which leads to overexpression of the protein. And it is very interesting that overexpression of MED13L protein causes much less trouble, and the patients are very mildly affected. There are, at the moment, 2 cases in the literature. One of them had a motor delay with walking at the age of 30 months, and which is not severe, and also had a normal speech. And the other case as well had some mild motor delay and mild speech delay. And this is also an interesting finding among the reported cases. I mentioned that seizures occur in 20% of the cases, and also another striking fact about the patients with seizures in the reported cases is the overrepresentation of missense variants in these patients. You can see here in this graph that 61% of those patients with seizure had missense variant, compared to 12% or 18% in the no seizure or not available groups. And this clearly shows that missense variant can lead more often to seizures in the patients. The type of seizures that have been reported can be classified into 4 groups. 32% of the cases had generalized seizures, meaning that almost the whole body kind of is involved in the process of seizing, with the average onset of seizures at the age of 87 months. Is quite a late onset. 17% of the cases had focal seizures, and focal seizures are the seizures that usually parts of the body are involved, and these are partial seizures. Average age of onset in this case is 42 months. 6% of the cases had febrile seizures that they are triggered by fever, with the average onset of 14 months. And in 44%, there is no specification about the type of the seizure. and the average onset of these seizures is 24 months. So this data basically shows us that the onset of seizure is not very early in MED13L syndrome if it occurs. But also we have really lack of data on details of the seizures. You can see 44%. There is no description in the literature, and this is a place also we need actually to have more information in the future. Also, about the treatment, successes and failures of the treatment, limited data is available at the moment, but at least there is one case with generalized seizures who responded well to orfuril or valproate or valproic acid, and one case with focal seizure who responded well with levetiracetam medication. The other feature which comes relatively in a higher number of MED13L patients are heart defects. 22% of the reported cases had heart defects. However, the majority of these patients with heart defects had septal defects, and these are usually holes between the walls of the two sides of the heart, and are often not significant, and they even can close spontaneously without any intervention. However, 35% of the patients have conotruncal anomalies. These are abnormalities in the outflow of the heart, and they certainly require surgery. However, with the advance of surgical techniques, this is also not a significant issue nowadays, and most of the heart defects can actually be repaired successfully. 9% of the cases also have valve defects. Another aspect of the features are abnormalities of extremities. These are also present in 50% of the patients. There is some sort of abnormality in the extremities, but 50% of these abnormalities, half of them are minor abnormalities in the finger or toe, and they require no intervention, and they are benign. 30% of the cases, they have ankle and foot deformities, such as, for example, clubfoot. You can see how it is in this figure, or metatarsus adductus. These are some sort of deviations of the feet, and they definitely need orthopedic follow-ups, and sometimes they need repairing surgeries as well. 35% of the patients also present with some sort of abnormality in eye and vision, but the majority of the cases is strabismus or crossed eye, which requires ophthalmological follow-up, and but there are often possibilities to correct them either surgically or without surgery. Another aspect of the syndrome which I want to introduce to you are the similarities of the facial features in the patients. This is not exclusive for MED13L, but for the MED13L cases, and there are some facial similarities which normally could help the clinician to diagnose the syndrome. For example, in 55% of the cases, there is this the bulbous nose. In 45% of the cases, there's hypotonic open mouth. The mouth is open due to the low muscle tone. In 25%, there are low set ears. The ears are located below this imaginary line, and as you can see, the features are really minor, and in a very low percentage of the patients, ptosis, for example, has been reported, which is falling of the upper eyelid. This may need intervention, or cleft palate also requires repair surgeries, but it's in a very low percentage of the reported cases. And of course, another important aspect for the children is the growth. And in this graph, I try to basically summarize the status of the growth among all the reported patients. Now you can see in this graph, here we have basically head circumference of the patient at birth and last visit. In the middle, in the yellowish color, is the stature, and here in the green is the weight. And on the y-axis here, you can see these numbers are standard deviations. And when the measures are above the 2 standard deviation or below the -2 standard deviation, they are considered abnormal. And you can see that although the average of the measurements are a little bit below the normal, which could be considered as 0, however, the majority of the patients are located between the 2 and -2. And therefore, we can say that growth parameters are not severely affected in MED13L patients. There are a few exceptions that, for example, the head circumference you can see here is above 2 standard deviation, which means it's large, or here is below -2, which is considered small head or microcephaly. The same applies to the stature, short stature, or high stature, and here is the low weight and overweight. But overall, the growth parameters are within the normal range. So by this, I would come to the summary of the presentation. I try to show you different types of variants which can affect genes, including MED13L. These are truncating, missense, and gain. For MED13L, the majority of the reported variants are truncating variants. Missense variants I showed you in that graph, they're clustered in some hot spots, which are very likely the important functional parts of the protein. Missense variants are more likely to cause seizures in the patients, and gains of MED13L are causing a mild phenotype. I also showed you from the pattern of inheritance that most of the variants occur newly in the patients, or they're de novo, but there are some reports of mosaicism, which means that some of the germ cells of the parents contain the variant. And speech is more severely affected compared to the motor development. Growth measures are not severely affected. Facial features are very minor, or facial dysmorphic features are very minor. And a percentage of patients have heart defects or abnormalities in the extremities or eyes, which may require surgery. By this, I come to the end of my presentation, and I would be very happy to take any questions. Thank you very much.
So my name is Randy Strick. I'm a professor in the Department of Molecular Biology at Rowan School of Osteopathic Medicine in South Jersey. And I'd like to thank the Simons Searchlight Foundation for allowing me to present my work today. And we'll be looking at the role of MED13L syndrome in mediating mitochondrial dysfunction. Now, MED13L and MED13 are part of a subcomplex called the CDK8 kinase module. And it's composed of 4 different subunits, obviously MED13 or MED13L, but also MED12 or MED12L, CDK8 or CDK19, and then cyclin C protein here. You can see they're all color-coded. And each of these proteins have a particular function in this complex. MED12 is a structural protein, as you can see, wanders through and sort of attaches everybody together. MED13 and MED13L are critical because they actually tether the CKM, the CDK8 kinase module, to the mediator of RNA polymerase. So this is an important function. CDK8 is a protein kinase, which means it attaches phosphate residues to other proteins to turn them on or off. And cyclin C is required for the activation of CDK8. Now, we've shown in my lab that this CKM module regulates about 3,000 genes and does so, it both activates them as well as represses them, depending on the particular gene in question. And today, although this is about MET13L, what I'm going to talk about mostly is cyclin C in this story. Now, the other thing I'm going to talk about are mitochondria. Now, everybody remembers from their 9th grade biology class that mitochondria are, say it with me, the powerhouse of the cell. And that's absolutely correct. The mitochondria produce ATP, which is the energy subunit for almost all the processes that require work in the cell. And without this ATP, when the mitochondria are this is when we're going to go back and start. So when the mitochondria are defective, things such as neuronal development and muscle strength are affected with reduced ATP production. Now, something else you probably learned was that they look like beans. This is an electron micrograph of cyclin C, or of mitochondria in the cell. But this is probably more of a due to an artifact of fixing these cells, because that's not really the way they look. In a normal cell, the mitochondria look like this. And these are now live cell images. And you can see the mitochondria are elongated and interconnected tubules. And this is actually very important because, and these are now mouse cells, and you can see that in general, in the normal conditions, the mitochondria are elongated. And this is important because when they're like this, they can produce ATP at a much higher efficiency. And also, very importantly, they can now repair any damage to the mitochondrial DNA or to the lipids that make up the mitochondrial membranes. So having the ability to fuse together is important for both production of ATP as well as repairing damage. Now, the mitochondria also can be seen in this fragmented phenotype. So when life is good, the mitochondria are like that, but there are conditions that reverse this ratio and make the cells produce this more. And one of those is under certain conditions of stress or cytotoxic drugs, the mitochondrial fragment. And this is the first step leading to a pathway called apoptosis, or programmed cell death. This is like a cellular suicide program that the cells undergo when they've been damaged too much. Now, somewhat confusingly is that mitochondrial fragmentation also happens during cell division. So when a cell divides into two daughter cells, they fragment the mitochondria to allow successful partitioning of the mitochondria to the individual daughters. And another time they see fragmentation is this quality control mechanism called mitophagy, in which damaged mitochondria are actually picked up and taken away for degradation. But you can see that this would pose a problem for the cells, because if it undergoes mitochondrial fragmentation, it wants to know whether it's doing it because of this, cell death, or doing it because of this, you're growing. And so that's where cyclin C comes in, that I talked about before. And these are now work done by a former graduate student in my lab, Kun Wang. And what he found is that, this is an assay I'll be showing quite a bit. This DAPI stain identifies the nucleus, and this red stain identifies the mitochondria. Now we're adding a third one, and this is identifying where cyclin C is in the cell. And under normal conditions, cyclin C is in the nucleus because it's part of the CKM regulating transcription. However, when you stress these cells for 4 hours by adding a pro-oxidant, what happens is the mitochondria take on this shape again. So here's the fragmented mitochondria we see in response to stress. But what you also see out in the cytoplasm are also these green dots, which represents cyclin C getting out of the nucleus. And if you zoom in on this merged image, you can see these yellow signals. And that's when the red from the mitochondria and the green from cyclin C overlap, you get yellow. And so that's something you'll be looking at later on as well. And so what we found is that this release of cyclin C from the nucleus is required for this mitochondrial fragmentation and also for efficient execution of programmed cell death. So that's how the cell tells between fragmentation that's involved in cell death and that involved in mitosis. So now you want to know where MET13 comes into this. And this is work done by a former graduate student, Svetlana. And what she found, and this is now in budding yeast, the organism. This is the well-known baker's, or maybe better known to some of you, brewer's yeast. And what we find is that in the same thing happens in yeast that happens in us, basically. Cytochrome C is a nuclear protein, and the mitochondria are elongated. However, what Svetlana did was she deleted MED13. So in yeast, there's only one copy of MED13, one copy of MED12. And so she deleted MED13, and what she saw was that cyclin C was no longer in the nucleus, it was out in the cytoplasm all the time. And that caused mitochondrial fragmentation all the time. As you can see here by the yellow signals, we're getting co-localization of cyclin C with the mitochondria. But what she found was that in addition to this fragmentation phenotype, she found that the mitochondrial function was absent in these cells, and mitochondrial DNA copy number was greatly reduced. And this is something that other people have seen before, where you get constitutive fragmentation of the mitochondria, like you see here. The mitochondria are no longer able to repair themselves, and you end up losing mitochondrial function along the way. So we published this paper in 2014, and a little bit later, we were contacted by a guy named Peter van Hasselt from the University of Utrecht in the Netherlands. And Peter is a clinician, and he had a patient that was identified with MED13L mutation. So this is the mutation. And typical of these MED13L mutations, he had primary phenotypes were heart developmental defects, moderate intellectual disability, hypotonia, and dysmorphic facial figures. And so Peter, because we'd published on MED13, he was interested in what we thought was going on with the MED13L phenotype. And so given our previous results that MED13 was required to anchor cyclin C in the nucleus, we said, well, if we can look at patient samples, we can see whether cyclin C is in the nucleus where it's supposed to be, or whether it's now out in the cytoplasm where it's not supposed to be. So we sent us fibroblasts from these patients, and so we did the analysis. And this is a control fibroblast, WI38s, they're normal, wild type. And this gives us the pattern we expected to see. Again, cyclin C is in the nucleus. This is what the DAPI is telling us where the nucleus is. And the mitochondria are elongated. So here's a zoom. Now, when we looked at the MED13L patient samples, we saw something very different. And that is, again, here's the nucleus, but look at where cyclin C is. It's always in the nucleus, but a lot of it is now out in the cytoplasm. And the mitochondria, instead of being elongated tubules, are now fragmented little dots. And you can see here in the blow up where there's fragmented mitochondria, and you see the yellow dots, that's where cyclin C is associating with it. Now this told us that there was cyclin C could be involved in mitochondrial fragmentation, but just because it's out there doesn't prove that. So we used this drug called perphytherin mu, or PFT, and we'd found out earlier that PFT stops cyclin C from getting to the mitochondria. And if you treat wild type cells with PFT, it doesn't make any difference because cyclin C is already in the nucleus. But we saw a striking difference though when we treated the mutant cells with PFT. And what you can see is in many cases, the cyclin C is back in the cytoplasm, or back in the nucleus, and the mitochondria are elongated again. So this told us that MED13L, one of the phenotypes associated with MED13L is this fragmented mitochondria, and it may be due, at least in part, to the aberrant localization of cyclin C. Now, as I mentioned earlier, several studies have noticed that constitutive mitochondrial fragmentation leads to reduced mitochondrial function. So we wanted to test that, and this is work done by a former postdoc in my lab, Kai T. And so he used what's called a seahorse analysis, and so he looked at mitochondrial function in the wild-type WI38s as well as the MED13L cells. And what he saw here was that the WI38s in blue, and this is the oxygen consumption, which is a normal measure of mitochondrial function. And this is what they found in the wild type cells. But in the MED13L cells, we saw a dramatic reduction in mitochondrial function. And if you look at ATP production, you also saw the same thing, where in the wild type, you got this level and you saw reduced levels in the mutant cells. Now, when we added PFT to the mix, we saw a partial recovery of both oxygen consumption and ATP production, but it wasn't back up to wild type levels. One of the problems with PFT, though, is that it is a bit toxic if you let it sit with cells too long. So we had to stop the experiments because they were getting the cells were getting sick. But one thing that Peter Van Hasselt had done with this patient prior was to get a muscle biopsy. And they looked at mitochondrial enzyme levels and all this. And this is 8 out of the 13 different enzyme reactions. As you can see, the patient values were lower than the normal levels in all of these cases. So taken together, these results would argue that, at least for this MED13L mutation, the mitochondria are exhibiting a high level of fragmentation, and they've also producing reduced mitochondrial output. Now, one of the causes for mitochondrial defects at this stage is the lack of mitochondrial DNA. So in the mitochondria, there's nucleoids. They're not very big, but they're called nucleoids. And here's a description of one. They code some of the genes involved in mitochondrial function. But there's anywhere from 500 to 5,000 of these nucleoid copies in a cell. So we wanted to determine whether the mitochondrial copy number was reduced as well in the MED13L strains. And it turns out, sure enough, it was. Using a PCR method, WI38 produced about 2,000 to 4,000 copies of the mitochondria per cell, which is pretty standard for a fibroblast. But the copy numbers in the MED13L mutant were only about 20 copies per genome, which is much less than this. But when you add PFT, you get a slight increase, but again, nowhere near what you would expect for the wild type cells. But one thing that you can do with this assay is determine whether the mitochondria are experiencing huge fragmentations in it, because this region here never gets deleted, and this region here often gets deleted. And so you can look at the ratios of these two. And it turns out that the genomes in the patient sample were not overly deleted compared to the control cells. So that's encouraging, and that says that we can maybe, if we can ramp back up mitochondrial function or mitochondrial DNA levels, we can maybe restore mitochondrial function. But so I mentioned earlier that of the roughly 1,500 proteins associated with the mitochondria, only a handful are encoded here. Most of them are encoded in the nucleus. So the next thing we wanted to do is see what MED13L mutation was doing to the expression of genes involved in mitochondrial function. And this is work done by a recently graduated student, David Stieg, and he looked at the role that CKM plays, and he came up with the number of about 3,000 genes. And of these 3,000 genes, a couple hundred of them are involved in mitochondrial function, as you can see with these GO terms they're called. So what he did was he selected 5 genes that were downregulated in cyclin C null cells, and looked to see whether they were also down in a MED13L fibroblast. And what he found was that all 5 of them did show a reduction in expression. Some's not so much, but others a little bit more. Anything below this line is a reduced transcription of these genes. So this now gave us the model that there's actually 2 arms of MED13L control for mitochondrial function. One of them is involved with these mitochondrial maintenance genes, we call them MMGs. And these are genes that are involved in mitochondrial function that aren't expressed as well as MED13 is involved with the morphology of the mitochondria as well. And so that led us to a model that with wild type MED13L, the CKM is intact in the nucleus, and it's promoting the expression of genes that are involved in mitochondrial maintenance. So these genes are being made, the mitochondria are fused, ATP levels are up, mitochondrial integrity is up, and the mitochondrial DNA copy number is up as well. However, in a MED13L mutation, we get partial release of cyclin C, which is causing mitochondrial fragmentation constitutively, and we get a reduction in mitochondrial maintenance genes. So this double effect here is causing reduced mitochondrial function, ATP levels are down, mitochondrial DNA levels are down. And the question now becomes, if this is correct, then can we maintain mitochondrial function or restore it by correcting the mutation, the MED13L mutation? So that's what we did next. This is the MED13L mutation, an extra T in exon 20 of MED13L. And so what we wanted to do was repair that, so get rid of that, put the wild type sequence back in. And to do that, we used a new derivative of CRISPR called ICAP CRISPR, which was developed by Ping Zhang, who's a collaborator of mine. And what Ping did was he developed a system in which he could rapidly replace the mutated copy of MED13L with a healthy copy. And we were very successful at that. This worked extremely well. And so to look at, once we got transfectants that had this corrected gene, we looked at, here's MED13L mutants, and you can see that cyclin C is out in the cytoplasm. The mitochondria are a mess, and you can see, as you can see here. But in our two corrected pools of transfectants, we see that cyclin C, the mitochondria are fine, cyclin C is in the nucleus where it's supposed to be, and everything has gone fine. So we've been able to correct at least the mitochondrial defect. And if we looked at another marker of mitochondrial function, which is elevated reactive oxygen in the cell, we saw a slight decrease in this, but it wasn't back to normal, where we hope to be. So that could mean one of two things. One is that we didn't wait long enough for the mitochondria to correct themselves, because these cells quit growing because they were, these are mortal fibroblasts, and so they have a certain lifespan and they quit growing. So this gave us hope that we're in the right direction, but we figured it's going to take a little bit more time to completely repair the mitochondria. So to do that, this is our future plans, is the generation of pluripotent cells, the iPS stem system. So we can take our both our mutant and corrected fibroblasts and turn them back into what's called stem cells, so that they can now divide forever, basically. And we can use that to determine whether we can recover mitochondrial function with the corrected mitochondrial cells. And this will give us a good idea not only what the extent of recovery is, but also the timing of recovery, because that's important as well. And the other thing that can be done with these iPS cells is they can be differentiated into different cell types, including neuronal cells. So now we can look at the impact of the mitochondrial dysfunction associated with the MED13L mutation on, say, neuronal development or muscle cell development or other types of cell types. And now that gives us a great reagent to look at a variety of different systems. And we're hoping at this time other researchers would be interested in this type of analysis. And one of the, and so sort of to sum up, one of the things that this meeting is good for is the ability to get people together that have been afflicted by this syndrome. And that tells you that, you know, there's a shared group of people out there to share these issues with. Now, it turns out that, again, we're with the CKM itself, and it turns out that the other mutations in the other CKM members have very strikingly similar phenotypes. MED13L mutation, MED13 mutations have heart defects and intellectual disability. MED12L, MED12 also have similar types of morphologies or phenotypes associated with it, as well as CDK and CDK19 all have these types of autism-like spectrum disorders. So all the mutations, all the genes involved with the CKM, when you mutate them, you get basically a continuum of phenotypes, except cyclin C. Now that's unusual, I think, because cyclin C, there's only one copy of it, whether there's two copies of the other proteins. And there's a couple explanations why this may be true. One is that you just can't mutate cyclin C all that well, and that's possible. But another possibility that we're going to look into is whether you need cyclin C to express the mutations that we find in these things, in these different genes. The idea being that if you make a mutation in, say, MED13, it won't be identified as, it won't be a disease-causing mutation unless it also allows cyclin C release, and that you need cyclin C out in the cytoplasm to affect mitochondrial function before you get expression of these different phenotypes. So in a future direction the lab is going to take, we're going to cast a wide net and look at mutations in these different factors. And it's a very simple assay for us. We'll just look and see whether cyclin C is cytoplasmic, whether the mitochondria are fragmented, and look at mitochondrial function in these different mutations. And this would maybe give us a larger family of mutations to work with. And this would do a couple things for us. One, it would attract more researchers into the field because there's other people working on these different genes. And two, it may raise the profile a little bit so the NIH would recognize it as a more common issue and perhaps be more likely to support it as well. So with that, I'd like to thank the Simons Foundation for allowing me to present and acknowledge people in my lab and collaborators. Katrina Cooper, who's a longtime collaborator, she's the one that actually found that the yeast cyclin C goes out and interacts with the mitochondria. Ping Zhang, who's developed the ICAP procedure for correcting MED13L mutations, and he'll be invaluable for doing both iPS cell production as well as making a MED13L mouse. And Peter Van Hasselt, who's been very kind to explain to me very slowly the intricacies of clinical diagnosis with MED13L, which as a basic scientist I didn't quite appreciate, and also current and former lab members and the support that's helped us throughout the time. So thank you very much for listening.